Human Genome Project is the 11th lecture which we will do in this particular series, that is molecular basis of inheritance. To start with, let us try to recall a little bit about the DNA. In a human cell, as it is shown in the slide, a human cell, it is made up of the chromatin material, which when condenses during cell division will condense or will form to result in 46 number of human chromosomes, as we are familiar with it. If extended out, the entire chromatin, it will extend to approximately 2 meters of length. The genetic material, the DNA content inside the nucleus, is in turn made up of 3 billion DNA subunits, that is the bases adenine, thymine, cytosine and guanine. There are 3 billion base pairs. Then furthermore, these base pairs, they walk in some units, those units which we call as genes, that help in coding for proteins through transcription and translation, as already discussed earlier. Now, it was estimated that it is approximately around 30,000 genes that are located within the DNA of human beings. Now, let us go to the Human Genome Project. Now, what is Human Genome Project? This was a project which was launched by the US Department of Energy and National Institute of Health in the year 1990. It took 13 years, 13 years to complete the project, that is 2003. But still, the one chromosome was left to be deciphered, but almost every work was completed by 2003. Here, the major contributions came from different countries like Japan, France, Germany, China, and many other such countries. Funding was done by Wellcome Trust of United Kingdom. Now, what does this project mean? This project is a project which was launched in order to understand the human genome, how the bases are sequenced. The number of bases, that is 3 billion bases, base pairs that make up the total human genome, total genetic material in a human cell, to understand how the bases are arranged. To understand that, this particular project was launched under the collaboration of different countries, funding by different countries. Of course, the major funding was done by the Wellcome Trust United Kingdom, and it was launched in 1990 and completed in 2003. It took 13 long years. So let us first try to understand what were the goals that was set to understand the project. The first goal, to identify all the genes, which was estimated to be around 20,000 to 25,000 in human DNA. It was estimated that there are around 20,000 to 25,000 genes in human DNA, and to identify all of the genes, what type of bases make up all the genes. Secondly, the goal was to determine the sequences of all the 3 billion chemical pairs that make up the human DNA. Human DNA, it is said to be made up of 3 into 10 raised to 9 base pairs. That means 3 billion chemical base pairs. To determine the sequences of all these base pairs, coding as well as non-coding. That was the second goal. Then also to store whatever information they can obtain by completing the project, to store everything in databases. Then whatever information is obtained, whatever knowledge is obtained by deciphering, by sequencing the bases, it can be transferred to other sectors like medical sectors, private sectors, where we can understand more about the diseases, we can understand more about the causes of diseases, cure of diseases, mostly to medical industry. Then finally, the one major goal of the project was also to address the ethical, legal and social issues that might arise from the project. That is, we are trying to understand the gene. That means we are invading into the privacy of a human, the genetic privacy of a human being. So definitely there will arise some ethical issues, some legal issues, even some social stigma associated with it. So to address all such issues was also one major goal of the project. So with all these goals in mind, the project was started in 1990. It is also called as a mega project. Why is it called as a mega project? Because one reason being the cost, because it was estimated in the beginning that to decipher to decipher, to understand one base pair, the cost will be $3 US dollars per base pair. So that way, if we are to decipher these 3 billion base pairs, then the cost would come to 9 billion US dollars in the beginning. But of course, the cost went far higher than that. So funding was a major thing here. So, and also furthermore, if the obtained sequences, when we can obtain the sequences, we want to store the information that is derived from them. We want to store it in the form of books. And if page of the book contained 100 letters and each book contained 1000 pages, then 3300 such books would be required to store the information. That is to make us understand the amount of information that is stored in the DNA, the genome of a human. So now the problem, and hence this was called as a mega project, cost as well as storing information, finding out the information, and then storing. So these were the as such it is called as mega project. And to overcome the difficulties that would have arisen in carrying out the project, of course funding from different agencies were done, and also bioinformatics landed a hand here, especially with regard to sequencing and storing. Now the methodologies that were applied. Two major approaches were applied. One was expressed sequence tags, which is abbreviated as EST, and the other is sequence annotation. Two methodologies were applied in order to find out or decipher the sequence of bases in humans. In the first, that is EST, the aim was to identify all genes that are expressed as RNA. As we are familiar that in the DNA, it has the total DNA material, it has both coding and non-coding regions, those introns and exons. So it has non-coding and coding regions. So in the first approach, first method, only the coding regions were taken into account to decipher, to find out only the coding regions. That will be the DNA that will express themselves as RNA. And in the second approach, second method, the sequencing the whole genome, coding as well as non-coding sequences, introns as well as exons. Two methods were applied. Now the genome annotation. Genome sequence can be classified into different groups based on overall sequence composition and structure. This is an overall view here that is given. Now the genome, it has coding and non-coding regions, exons and introns. Now the coding regions, they are simple compared to the non-coding regions. Coding regions are the genes that we talk about, and these genes will express through transcription forming RNA and then translation to form the protein. So finally these genes will be expressed as proteins or RNA. So the first approach, that is expressed sequence tags, followed this approach. That means what was done? The RNA was studied that will be present in a cell, and then the RNA was made to undergo reverse transcription where we will get the DNA, and then accordingly we can find out the sequences. So it was focused only on the coding part. Now coming to the non-coding part. Now the second approach, sequence annotation, it took into account both coding and non-coding regions. Now the non-coding regions, that is the introns, they were of two types: introns, okay, regulators, etc., which will not code for anything, otherwise also they will not code for anything, and a portion was called as, we name it as repetitive DNA. Repetitive DNA means some sequences of DNA, some base pairs of DNA which gets repeated continuously. Which gets repeated continuously when we call it as tandem repeats. They were further called as satellite. They can be two types, mini satellite and microsatellite. We have slides, I have slides over here where I'll be discussing about it. Then another type of repetitive DNA where some sequences they're repeated but interspersed. That means they're not continuous. These are transposons. These are slightly beyond the topic of discussion for us at this point of time. We will be talking about the tandem repeats, which was actually focused while carrying out the Human Genome Project. First, express sequence tags. Now, as I have already mentioned, express sequence tag was what was done. The tissues were taken. From the tissues, the mRNA that will be present in the tissues that were located out, through reverse transcription, they were made the DNA from that RNA, and then the DNA were tagged. DNA was understood, that the sequence of DNA was understood. So coding regions of DNA genes were mainly studied here. RNA is isolated from tissues. RNAs are reverse transcribed to DNA. DNA is fragmented. The DNA that will be obtained will be fragmented, means it will be cut into small pieces using the enzyme endonucleases, and that will be then separated out and finally sequenced. So this was the approach for expressed sequence tags. Now, before going into the second approach, first we will like to, it will be better if we understand what is satellite DNA. Now, satellite DNA are short sequences of DNA which gets repeated. Like suppose ATGC. Again, the next 4 bases also ATGC. Again, it is ATGC. Again, ATGC. This is an example I'm giving. So it gets repeated one after another continuously. When it is continuous, we use the word tandem. Short tandem repeats that consist of 1 to 9 base pairs. 1 to 9 base pairs. So it may be one pair, like say 80, 80, 80, 80, or maybe 80 GC, again 80 GC, again 80 GC, or it may extend up to 9 base pairs. If it is so, then we will refer to that fragment or that fragment of DNA as microsatellite. If the repeats, the length is more than 9, the length of the base pairs, it is more than 9, it is 10 to 100. Like say ATGC, ATG, it's a, that way the sequence, it goes to 10 base pairs, 10 to 100 base pairs. Again, suppose it is made up of 50 base pairs in a sequence. Again, the same 50 base pairs are repeated. Again, the third time also the 50 base pairs get repeated. Then such type of repeats will be referred to as mini satellites. Usually it has been observed that microsatellites are rich with adenine and thymine, whereas the mini satellites, they are rich with guanine and cytosine. They are also known as simple sequence repeats, and these are called as variable tandem repeats, VNTR. VNTR we will again talk about when we do DNA fingerprinting. This has a very important application in DNA fingerprinting. So here we have the chromosome, the DNA. Within the DNA, some repeats are there. Like say this particular portion is repeated after certain interval. Okay, they are repeated here. Then again they get repeated. So when it is 1 to 9 base pairs, microsatellites, 10 to 100 mini satellite. If more than 100 base pairs, then it will be called as macrosatellites. So here it's an example I've given here so that we get a better concept of satellites. Like say individual 1. There are the 2 individuals. This is the DNA of the individual. The total DNA of individual, not a portion of the DNA, I can say. Okay, so here the person had GGT. It had a base. These are the bases that were present. Here we are showing only one of the strands of DNA. Now here the bases are say AGTC, GGTAAG. Same repeat, if you can see here, the same repeated, the same base pair is repeated, again it's repeated, it's repeated. So how many times? 1, 2, 3, 4, 5, 6. 6 times these base pairs have been repeated in individual 1. In the second individual, base pairs remain the same, but the number of repeats vary. 1, 2, 3, 4, 5, 6, 7, 8. So we can clearly see here the number of repeats have changed. Only this portion, the bases are similar in both the two cases, but the rest of the base pairs, see, the rest of the base pairs are not the same. This is unique to the person. But the base pairs here, the portion which is getting repeated tandemly one after another, such portions are called as satellite DNA, repetitive DNA, which will, the number of repeats will vary from individual to individual. Okay, the type of mini satellite, because the repeat sequence is 10 to 100 nucleotides here. These nucleotides, the number of these, how many? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 base pairs. So this is a type of mini satellite. If it was less than 10, then we will call it a micro satellite. The sequence repeats 5 to 50 times. So this number of repeats can extend from 5 to 50 times, and it differs from individual to individual. But the repeating sequences does not. Okay, so the repeating sequence remains the same. This sequence remains the same, but the number of repeats will vary from individual to individual. Again here, just to make it more clear, there are 3 individuals given here. The number of repeats, say AG80. AG80, this is site 1, short tandem repeat. This is second region, short tandem repeat. Within a DNA, there can be different repeats, different like sites. So the particular sequence here, AG80. So this is a very short sequence, less than 10. So we can call it as a micro satellite. How many times? 7 times it has been repeated. Again, in another portion of the same person's DNA, it has been repeated 6 times. Second individual, location is the same, but number of repeats is less. Location is the same, type of repeat is same, the sequence is same, but the number of repeats is different. Accordingly, the third. So such type of repetitive DNAs, these are called as, these are non-coding regions. These are always non-coding regions, and they are very unique to the individual too. So the satellite DNA, as well as this, that which will include the non-coding regions, and these are the coding regions that are given here. Coding regions, everything was deciphered using sequence annotation. Now, in the method of sequence annotation, what was done? The DNA is extracted from the cell, then it is fragmented, it is cut into small pieces using an enzyme called as endonuclease. Then these small fragments, they are cloned into vectors. Vectors means are plasmids. Usually plasmids are used here. Plasmids are used here. Plasmids, that is bacterial plasmids, they are used here, which will help in transfer. That means this small fragment of human DNA is joined with a bacterial or yeast DNA, and then it is inserted into the microorganism. Let the microorganism divide. As it divides, what will happen? The number of copies of the human DNA will also increase. So thereby, what are we doing? We are increasing, we are cloning. We are cloning the human DNA, that small fragments of DNA, into many number, to increase the number, to make it multiple copies. After which they are again sequenced. Okay, they are again taken. Okay, they are digested, and then they are sequenced in the library. That means some sequence annotators are there, already prefixed are there. They are compared, and then we can sequence the DNA. Then after which they will be assembled and as contiguous fragments. Now, assembling part, the involvement of satellite DNA comes during the assembly, because as we have seen, the repeats, the location is the same from individual to individual, just the number of repeats will differ. So that will help us in assembling the DNA again, which we had already fragmented, inserted into different bacteria, allowed them to grow into multiple copies again to assemble them, it is the repetitive DNA will help. So here what was done? Coding and non-coding both regions were sequenced. The steps we repeat here. DNA is isolated, it is fragmented using endonuclease, it is cloned to produce multiple copies. Usually the two commonly used organisms were bacterial artificial chromosome and yeast artificial chromosome. These were taken as the hosts into which the human DNA was inserted. Then finally, it was sequenced using automated DNA sequences which were developed by Frederick Sanger. In a different presentation, in a different lecture, I'll be talking about the Frederick Sanger's automated DNA sequencing, the procedure, the principle or the procedure that is used. Here for the time being, we can simply learn it this way, that after they were multiple copies were produced, then they were sequenced using the automated DNA sequences. Then when all the methods, whichever method was followed, after completing, the final outcome of the Human Genome Project was put under different headings. Human genome consists the conclusions. That means the human genome consists the information of 24 chromosomes. Why 24? 22 autosomes plus X chromosome and also one Y chromosome in males. Otherwise, we have 23 chromosomes, 23 pairs of chromosomes, which makes 46. That means an individual may have 22 autosomes plus X or 22 autosomes plus XX or 22 autosomes plus XY. So all of them were deciphered. We have been able to understand all the chromosomes, the DNA sequences in all of them. The human genome contains over 3 billion nucleotide pairs. Human genome is estimated to be having about 30,000 genes. 30,000, which was earlier thought to be only around 25,000. Average gene consists of 3,000 bases. 3,000 base pairs on an average makes up one gene. But this varies, of course, greatly. It is an average value that is taken, and the largest gene is dystrophin, which is made up of 2.5 million base pairs. So we can see here, out of these 3 billion nucleotide pairs, 2.5 million makes up a single gene here. But the fact is that only about 3% of the genome encodes amino acid. So out of the total genome, we have 3 billion nucleotide pairs, but out of that only 3% helps in formation of amino acids and proteins. The rest of it is simply junk, that is the repetitive DNA, what we saw a little while ago, only repetitive DNA. Functions are still unknown. We are yet to find out all the functions of the genes. Then the repetitive sequences make a very large portion of the human genome, have no direct coding function, but of course they do not help in formation of any protein, but of course they tell us many things about the evolutionary trend. Chromosome number 1 has the most genes. Highest number of genes is present within chromosome number 1, and Y has the lowest. It is the smallest chromosome and 1 is the largest chromosome. All nucleotide bases are same in all people. In all of us, the bases are the same. Those repetitive DNA, the number of repeats are different. That is, other than that, we are talking about the coding regions. So all nucleotide bases are same in all the people. Okay, what differs? Okay, genome sequences are different, differ only for less than 0.2% of base pairs. So we differ only in 0.2% of base pairs. Otherwise, all the base pairs of all the humans on this earth are the same. And those differences, okay, the differences in DNA due to the position of the bases, some bases they will differ only at certain points. Some bases will be different between two individuals, among three individuals, and that creates the different types of DNA. which we call as DNA polymorphism. Now, one important point to learn here is single nucleotide polymorphism. That means when there is a difference of only one base pair between the DNA of two different individuals or among more than two individuals, then we will refer to that sequence as single nucleotide polymorphism, single base differences, also called as SNPs. In short, we call as SNPs. One SNP occurs at every approximately 1000 base pair of human genome. So, 1000 base pairs are similar between two individuals. Then after the 1000 base pair, there will be one difference. If in one person it is A, the other person it might be G. So there the difference will occur. 85% of all differences in human DNAs are due to SNPs. So we are all unique. We all are unique. Why are we unique? Only due to this SNPs. The single base difference creates so much of differences among individuals. Throwing little more light on single nucleotide polymorphism, SNPs. These are positions in a genome where some individuals have one nucleotide, example guanine, and others have a different nucleotide, example cytosine. Okay, so here you can see these are three people shown here. All the base sequences, they are the same except this location. The first person it is A, second person is G, third person is T. And due to such differences which may occur again after a certain length of the DNA, due to these differences only we are all unique. We are unique with characteristics which are which makes us different from others. Okay, so although each SNP could potentially have 4 alleles because there are 4 nucleotides. Okay, so this is about diseased conditions. Okay, this is about diseased condition. Then polymorphism, poly or many morphoforms. So many forms in a general population. Okay, this particular base pair is different. This particular base pair is different. Otherwise, everything remains the same. Okay, and similarly here also you can see this base pair. See T. Okay, G, general population, they are having 94% of the population they are having the base pair G. 6% they are having T. Single nucleotide polymorphism. So at one base it is differing. Or it may be other way around, like say there will be more people, like say in a third person again it might be C, and at the fourth person it might be A. So they will differ in this way. Again, single nucleotide polymorphism, here usually called chip sequence or SNPs, SNPs. 3 people having the DNA sequences here where they differ only at one base. Otherwise, the DNA remains the same but they're differing at one base. So this is known as single nucleotide polymorphism. Poly means many, morphism forms. So one base pair differences gives us different forms. So all these information, all where could be gathered or could be understood after sequencing the entire human genome. Next, we will be moving over to DNA fingerprinting and also to the Sanger's annotation technique. Thank you.